Hi students. So far you are learning about catalysis and its types of catalyst and how a catalyst action is performed by studying various theories. And this module you're going to discuss about enzyme catalysis. Enzyme is a biological catalyst. Every biological system, so many biochemical reactions are taking place which are catalyzed by enzymes. Enzymes are complex protein molecules with three-dimensional structures. So enzyme is a macromolecule. It is a protein molecule. It has three-dimensional structure. They catalyze the chemical reactions in living organisms. They are often present in colloidal state. They are neither true solution nor a suspension. Generally, every protein is a colloid. So like that, enzyme is also exist in colloidal state. And it is extremely specific in action. Specific, the term suggests a particular enzyme is used for catalyzing a particular biochemical reaction. How? Finely divided nickel is used for hydrogenation of oils. Like that, a particular enzyme, for example, glucose forms a polymer called glycogen and stored in the liver. This glycogen preparation is called glycogenolysis. This glycogenolysis is catalyzed by a specific enzyme called glycogenase. It cannot be catalyzed by any other enzyme. Like that, this enzyme cannot catalyze any other biochemical reaction. So that is how an enzyme is said to be specific in its action. This graph you have already seen while discussing about catalysis. This graph once again shows the difference between uncatalyzed reaction and catalyzed reaction. In the core, the graph is drawn between the course of the reaction and free energy. Course of reaction, how the reaction proceeds, is taken in the x-axis and free energy is taken in the y-axis. The reactant energy is mentioned in the graph. The reactants directly will not give the products. It reaches an activated state, and this state is attained by the reactants by gaining some energy, and it reaches a maximum energy. The point where the energy barrier is kept is called the threshold energy, and once it reaches the threshold energy and it forms the products, you will be able to see the the portion of energy between the reactants and the energy barrier, that is the threshold energy. This energy is called activation energy. See the second graph. In the second graph, the graph is drawn for a catalyzed reaction. Here, look at the energy barrier. The reactants need very minimal energy in order to reach the energy barrier to form the products. So activation energy is more in the case of uncatalyzed reaction, that is the energy consumed by the reactant is more in the case of an uncatalyzed reaction. Here in the catalyzed reaction, the energy consumed to reach the threshold energy is very less. So activation energy is more in the uncatalyzed reaction, activation energy is less in catalyzed reaction. This is the difference between an uncatalyzed and catalyzed reaction. Now you can understand the importance of a catalyst which is used in a reaction. Some examples for enzyme catalysis, that is reactions that are catalyzed by enzymes, biological catalysts. Cane sugar, how it is converted or decomposed to glucose and fructose. Cane sugar is nothing but sucrose, which are manufactured from sugar cane. So it is called cane sugar. It is hydrolyzed in the presence of the enzyme invertase in the acid medium to form glucose and fructose. This invertase enzyme is used only for this purpose. The second reaction, how glucose is converted to ethanol. And these two reactions are the reactions that are taking place in the manufacture of ethanol from molasses. And the third reaction, how starch is decomposed to maltose. The enzyme which is specific for this purpose is diastase. Starch is a polymer, so it is mentioned as C6H10O5 N times. With water, it is converted, polysaccharide is converted or decomposed to disaccharide maltose. And then how maltose, in the presence of the enzyme maltase, is converted to glucose. Maltose is a disaccharide which contains two glucose units. Like how sucrose contains one glucose and one fructose, maltose contains two glucose units. For this, the specific enzyme which is used is maltase. These are certain reactions which are catalyzed by enzyme. All these are biochemical reactions. What are the characteristics of enzyme? We are also going to discuss about the characteristics of enzyme catalyzed reaction, the characteristics of enzyme catalysis in detail. But here we are going to study about the nature of enzymes, what an enzyme is. Already we have discussed certain points under this. It is a protein. It catalyzes biochemical reaction. Without enzyme, the biochemical reactions will be very slow or it may not take place. Every reaction has its own specific enzyme. Some enzymes are globular proteins. Most of the enzymes are globular proteins. A few may be made up of RNAs, ribonucleic acids. Enzymes undergo all the reactions of proteins. Enzyme is affected by temperature and pH changes. Enzyme activity is usually regulated by other substances in the cell. These are the characteristics of enzyme. What is an enzyme? What is a catalyst? Enzyme is a type of catalyst, we know. It is a biological catalyst. What is a catalyst and enzyme? What is the difference between the two? Catalysts are substances that increase or decrease the rate of a reaction but remain unchanged. Enzymes are proteins that increase the rate of the reactions, converting substrate into product. So enzymes in biological system are usually used only to increase the rate of biochemical reactions taking place in organisms. Catalysts are low molecular weight compounds, whereas enzymes, they are globular proteins, so naturally they will have high molecular weight. There are two types of catalysts, positive and negative. There are two types of enzymes, activation enzymes and inhibitory enzymes. Activation enzymes helps a reaction to take place. Inhibitory enzymes deteriorates a reaction, means if a biochemical reaction should not take place in our body, that reaction can be prevented by by having inhibitory enzymes. This slide shows how an enzyme reacts. Here the reactant is called as substrate. E stands for enzyme, S stands for substrate, which is a reactant. And the enzyme has active sites or active centers where the substrate molecule can come and bind. You could see the diagrammatic representation of enzyme and substrate. So it is like a lock and a key. This enzyme and substrate forms a complex ES, which is called activated complex or enzyme substrate complex. Now the substrate is ready for reaction. So the first step is called binding step, where the substrate binds onto the enzyme. Second step is called catalyzed step, where the enzyme substrate complex decomposes to enzyme and products. This hypothesis, what we have studied previously, is called the lock and key hypothesis. The lock and key hypothesis says that, like how every lock has its own key which can open, unlock a particular lock. Like that, every enzyme is unique and specific for a reaction. So in the first step, again the diagram shows the substrate binds to a free enzyme. The enzyme is the blue color structure over there, and the substrates are green and yellow structures, and how the enzyme substrate complex is formed, and then how the product is formed and it is desorbed from the enzyme. So that is very well explained in the picture. Now we are going to study about characteristics of enzyme catalysis. You need to remember about 6 important characteristics of enzyme catalysis. The first one, highly efficient. The enzyme catalysis is highly efficient. That means a small amount of enzyme, a pinch, 1 microgram quantity of enzyme can be used to catalyze reactions involving millions of substrate molecules. Highly specific. This we have learnt enough. That is, a particular enzyme is used for a particular biochemical reaction. A particular enzyme catalyzes a particular reaction. And third point, highly active under optimum temperature. See, optimum temperature is the temperature at which the activity of the enzyme is maximum. Enzyme helps the substrate to form products, and this can take place at different temperature. You all know our body temperature is 98.4 degree Fahrenheit or 37 degree Celsius. Only at this temperature the activity of enzyme is greater in human systems. Below or above the temperature, the activity of enzyme will be disturbed. At high temperature, when we have fever, the activity of enzyme is very much deteriorated. Hence, a person may feel less energetic when he or she is having fever. Above the body temperature, the enzyme may get denatured. After all, it is a protein. So it gets denatured into strands of proteins, and that's why it could not take part in the biochemical reaction. It cannot help in the biochemical reaction. The fourth point, highly active under optimum pH. The activity of enzyme is also responding to the pH. At a particular pH, the activity of enzyme is maximum. So that pH is termed as optimum pH. We are going to discuss about this characteristics in detail. Increasing activity in presence of activators and coenzymes. Activators and coenzymes are substances which can increase the activity of enzyme. Like how the promoter increases the activity of a catalyst, like that activators and coenzymes can increase the activity of enzymes. Influence of inhibitors and poisons. Inhibitors are substances which will inhibit the activity of enzyme. We'll discuss about certain points under this characteristics in detail. Specific is the second point in the characteristics of enzyme. The properties of enzyme. Specificity of enzyme is explained here. There are various types of specificity. You need to know about the first type. That is, one enzyme acts only on one substrate. Urease is an enzyme that decomposes only urea. It cannot decompose glucose. Arginates, Arginase is an enzyme that splits only arginine. It cannot split alanine. So like that you have different types of specificity of enzyme. The other two types are relative specificity and stereo specificity. So we, we need not know about these types of specificities, but still we can understand what it is. Relative specificity. One enzyme acts on different substrates which have the same bond type. See, certain enzymes can act on different substrates, but the basic structure of those substrates or the bond formed, the enzyme is going to break, is the same. See, for example, in proteins are made up of amino acids. Amino acids combine with each other to form a polymer called peptide. Peptide forms polypeptide. Polypeptide forms proteins. The basic bond lying between the amino acids in protein is the amide linkage. The amide linkage is called CONH bond. The CO comes from the acid group, NH comes from the amine group. So the NH linkage is broken by the enzyme pepsin. So pepsin can be used for breaking or dissociating or splitting any type of protein, because after all it is going to cleave the same type of bond called the peptide bond or the amide bond. Stereospecificity. Some enzymes can catalyze the transformation of only substrates which are in certain geometrical configuration. So there are two type of configuration, cis and trans isomers. So if a particular enzyme can catalyze only cis isomer, it is very specific that can catalyze only the cis isomer, not the trans isomer. Next, how temperature affects enzyme activity. The graph shows the temperature in the x-axis and the rate of the reaction, the velocity of the reaction in the y-axis. As the temperature increases, the heat energy increases, so more collisions between the enzyme and substrate, but it reaches a maximum. Optimum temperature for humans is close to 37 degree Celsius or 98.4 degree Fahrenheit. And beyond that temperature, the activity of the enzyme is decreased. The rate of the reaction decreases. This is due to the denaturing of enzymes at high temperature. So the rate of the reaction falls rapidly. Effect of pH on enzyme activity. This pH graph, that is the graph which is drawn between pH and enzyme activity, shows that optimum pH at which the enzyme activity is maximum. The rate of enzymatic reaction depends on pH of the medium. Each enzyme have the pH where the enzyme is more active, and this pH is known as optimum pH. For most enzymes, the optimum pH lies between 5 and 9. And then we were discussing about activators and coenzymes. Activators help as well as coenzymes help to activate enzyme. Activators can be cofactors, that is, they are non-protein small inorganic compounds and ions. Coenzymes, non-protein organic molecules, they bind temporarily or permanently to enzyme near active site and enhances the reaction. Example, many vitamins can act as coenzymes. can see in the picture how the enzyme activators work. Activators first bind on to the enzyme near the active site, modify the active site for enhancing the binding of the substrate onto the enzyme. So without activators, the binding is not easy. This is how the enzyme activators work. Then enzyme inhibitors. An enzyme inhibitor is a compound that decreases or diminishes the rate or velocity of an enzyme catalyst reaction by influencing the binding of substrate and its turnover number. The inhibitor may be organic or inorganic. Usually the drugs, antibiotics, toxins, antimetabolite or natural products of enzyme reaction can act as inhibitors. How the inhibitors work? See, look at the first picture without inhibitor. The substrate binds onto enzyme and then forms products and desorbs from the enzyme. Here, when the inhibitors are there, the inhibitors bind themselves at the active sites and there are no active sites for the substrates to bind. This is how the activity of enzyme is inhibited by the presence of inhibitors. They will block the active sites and the active sites are unavailable for the substrate molecules. So that is how the reaction is disturbed. Enzyme activator, enzyme inhibitor, the difference between the two. This is just for our interest. We are just comparing the activator and inhibitor. Enzyme activators are chemical species that can bind with an enzyme to increase its activity. An enzyme inhibitor, opposite, to decrease its activity. Enzyme activator can increase the activity of an enzyme, decrease the activity of the enzyme. Again, the same point repeated. And third one, example. Example for activators, hexokinase 1 and glucokinase. Inhibitor include drugs, ribonuclease inhibitor, etc. Inhibitors, for example, sulfur drugs, penicillin drugs, they deteriorate the activity of a certain bacteria in our body. So these drugs can act as inhibitors because when the bacteria gets multiplied by with the help of our cells, or they multiply by themselves, in order to prevent the multiplication of a particular germ or a bacteria, this antibiotics are used. How the antibiotics act? They act as inhibitors. See, when a bacteria is going to multiply, biochemical reaction must take place. An enzyme must help for the reaction. This enzymic activity is inhibited by the help of these drugs. So naturally, the reaction gets disturbed. And this is how the drugs work in our body. So inhibitors are a very important part in the pharmacological field. Thank you, children. I'll catch you up in the next video with another concept.